right, well, thank you. And I was going to make a correction. It's Quartic.ai. My apologies for missing that one. So with that, Brian, take it away.
Okay, great. Thank you very much. Yep, so good morning, everyone. My name is Brian Pope, Solutions Engineer with Quartic.ai. And I know I'm the last thing standing between you and lunch, so I'll try to keep this relatively brief. And Quartic, just so you all know, it's an industrial AI and data platform specifically designed for pharmaceutical manufacturing. And what I want to talk today is about how we can leverage things like AI optimization, automated PAT, digital twins to help enable continuous biomanufacturing and at least take the first step towards that if you're not ready to do it for like a whole plant or something like that. So we'll get into kind of why move to continuous, some of the requirements and challenges in doing so, how Quartic can help with that. I'll go into an actual demo of how we've done this for a single unit, specifically a continuous bioreactor for penicillin production, and then how we can expand that to a larger system, a complete end-to-end digital twin for continuous mAb manufacturing. So if we start with some of the benefits of continuous biomanufacturing, hopefully being in this track, you guys have heard some of this already from earlier presentations, but better flexibility and adaptability, being able to adapt to changes that are happening in your process, especially using some of the technologies we can use. Faster speed, lower cost, lower materials. You can eliminate process unit hold times between them. You can eliminate some of the hold times between different batch runs, cleaning phases, all of that. We can also reduce the overall equipment footprint because of the continuous manufacturing, reducing that time. We can use less equipment or have more overall throughput. In terms of quality, there's been studies that have showed in continuous manufacturing, the final product quality is at least as good as from traditional batch-based processes. And being able to eliminate a lot of the human interactions, potential human error, we could get even a little bit better. Then in terms of availability, because the cost is lower, the speed is higher, those cost savings can be passed on to consumers, to the patients, making the products more broadly applicable within the marketplace for everyone. So if there's all these benefits, then the question is, well, why isn't everyone doing continuous biomanufacturing? Why isn't everything done this way? And the answer is because there's a lot of challenges still there and certain requirements in order to be able to do this. So if we think about processes that have been made continuous, things like small molecule chemical API, formulation, packaging, a lot of times those processes are a little bit less complex, and also on the time scale, usually they take less time. So if we think about something like granulation and tableting, you have roll forest, compaction forest, maybe moisture content, and tablets are made, in an order of a few minutes, something like that. So if you lose a few tablets, a few hundred tablets, it's not that big of a deal, versus if you lose a 20,000 liter production reactor that's been running for 3 weeks with $1 million of media in it, that's probably going to be a bigger deal. So there's a level of complexity there, and also just in terms of what you end up monitoring, right? For large scale reactor, you have pH, temperature, agitation rates, sparger rates, what's happening with the cells themselves, death rates, growth rates, things like that. So there's a higher data requirement there. So we can't be relying on paper systems. Everything needs to be digitized, and you need the equipment and the systems to be able to support that, including the software systems. We also need to get some sort of real-time quality feedback. So ideally inline sensors like Raman probes, NIR, IR probes, and then be able to add analytics on top of that to automatically turn that into quality attributes, things like, what's my current concentration or yield? And then once you get to that point where you can say, all right, in real time, this is kind of what it looks like inside of, say, my reactor. Then we need to be able to characterize that as, is this good or bad right now? And can I predict this where at the end of my process, am I going to have good or bad yield? If it's going to be good, can I make it better? If it's bad, is there something I can do to fix it? So that's where we start getting into the predictive modeling, prescriptive analytics, and optimization. But some of the challenges there, it kind of comes back to the data piece. Oftentimes, in order to build these machine learning models, a lot of traditional methods, you might need years worth of data, a large, perfect, clean data set that doesn't exist, especially if you start talking about new products, new modalities. So one thing here is if we can get to a more sample efficient approach to machine learning that requires a smaller data set, that's kind of the ideal scenario that we have to move towards. And then finally, one of the biggest hurdles of all might be getting kind of over the trust factor. Humans have to be able to accept this, realize that it might be better than the way they've already been doing it. Does it make their lives easier? Can they trust the output of the ML and AI algorithms or whatever other systems and calculations are being used within this? So oftentimes it's kind of the biggest hurdle, getting people to accept change and move on to this new method be of continuous when they've been doing batch-based processing for decades. So with that, just because it's challenging doesn't mean we shouldn't be getting started with this today. So how can we get started? What are some of the things we can do? Well, on the previous page, if you think about all the different bullet points there, each one of these traditionally might be a separate system that you're using, right? You might have a DCS system or a historian for the data, another one to add context on top of it, another one to do that inline monitoring, something else for predictive modeling, something else for optimization. So we start talking about 5, 6, 7 different systems. They all need to be installed, maintained, and integrated together just to get off the floor for doing this continuous manufacturing. So if we can simplify this down to 1 or 2 systems, and me being from Quartic, I'm a little bit biased. Quartic can do that. But even if you don't use Quartic, if you can find some other software package or systems that can do the data piece, the contextualization, add in the analytics for real-time and predictive, be able to optimize set points, do right back down to PLCs and controllers to get into autonomous control. These are kind of some of all the different pieces that ideally you want to bring together in order to enable this continuous biomanufacturing. The other piece is, unless you're doing something completely greenfield, you're probably not going to apply this across an entire plant all at once. That's just a lot to do with new technology, new systems. So pick one unit that you can start with. So I'll show an actual demo of what Quartic has done with the continuous penicillin bioreactor. And then from there, we can expand out into multiple units, multiple process areas, and eventually an entire plant. So to start off with what we've done for kind of the single reactor scenario. So down in the bottom right here is the continuous reactor where we're pulling in sensor data, so temperature, DO, pH. And then we also have an inline Raman Pro getting spectral data out, and we can handle both the streaming tag data, the sensor data, and also the spectral data natively, and then run a model to turn that spectral data into real-time quality results as well. So from there, so now we have the inputs, which are these process parameters we're controlling, and the outputs, which are those quality attributes. So build a machine learning model to understand those relationships. So again, if we see, depending on what we see, the quality coming in as what changes can we make, what adaptations can we make to make this quality better. And we're going to continuously feed data in this. It's all live streaming data. This isn't something we're doing, once a month or every 6 months or something. It's continuously updating. So every new iteration we're running, every bit of time we're running in this reactor, we're getting more and more information, more data that we can use to retrain the model and get better and better with time. And then finally, the last piece is getting into the optimization piece, moving towards continued AI-driven control, where we can actually make set point recommendations based off of this. You'll see here we talk about a small data AI-based algorithm. So rather than needing years of data, Quartic typically we can run with about 10, I'll say, batches worth of data. So this works in continuous, also in batch-based process. That's where we've done most of our actual implementations. And after about 10 or so batches, we can be giving very accurate set point recommendations. When we move into continuous, oftentimes there's still an idea of kind of an artificial batch for like quality control purposes, where we're saying batch 1 starts and stops here, batch 2 starts and stops here, for tracking genealogy, those types of things. So we can still use those batches as a concept to iterate over, or the other option is to do it more time-based. So if you make some set point change, how long does it take us to see a change in the quality? Is it 5 minutes? Is it an hour? Whatever that time period is, we can use that as an iteration that the AI uses to iterate through and learn. So kind of the outputs of this, have a model that understands the relationships between the process parameters and the quality attributes, be able to predict the quality and performance of the overall process. In this case, we'll focus on yield specifically, and also be able to make recommendations for optimized set points. So just to kind of show what this optimization application actually looks like, we'll run a video here. All right. So again, we're focused on penicillin fermentation here and continuous reactor. And just by choosing penicillin up front, it's going to automatically tie into the platform, pull in full recipe information, what parameters are we controlling. And And then at the top, we're going to choose what are we trying to optimize. In this case, our objective is maximizing yield, but if there's some other quality parameter, maybe it's like biomass evolution time, maybe it's residence time, purity, minimizing impurities, whatever it is you're trying to optimize, you select that. Then in this list at the bottom of the recipe, we have a starting set point value from the recipe and also a range that the optimizer is allowed to adjust between, so upper and lower bounds. And this is shown visually over on the right-hand side with that dark orange line. This is my set point from the recipe, and then there's a lighter orange band, which might be a little hard to see, but that's the upper and lower bounds that the optimizer is allowed to adjust between. So we're not stuck with just a single set point, but we can optimize within a range that's been identified as safe in order to get to an acceptable final product quality. And the thing here is this is actually all we need in order to build this AI and this optimization algorithm. We don't need a lot of historical data. We actually need no historical data in this case. We're running just off of that recipe as a baseline. And then we'll iterate through and learn as we do live runs. If you have historical data, great, we can pre-train the model and you'll get optimized set points faster. But in this case, we're starting with nothing. So as this runs, we'll be able to see set point recommendations that come out over here. At the top, we have a chart showing the yield overall for each individual run that we're doing. And we're not hooked up to a live process here. That's how we're kind of able to iterate very quickly. Instead, we have a high fidelity penicillin digital twin that's running, that's able to generate data around the pH and the temperature and the Raman spectroscopy. So we're generating, in this case, we're using that digital twin just to generate simulated data from the production process. We feed this back into the AI algorithm. Based on the new data, it reruns and says, okay, these are the new set points for the next run we want to go through. It feeds that back into the digital twin, it generates data, and then it cycles through. So by doing that, we're able to quickly go through 10 or so runs without, whereas normally this process might take 200 hours to do just a single run. So in this case, the digital twin is really running just to provide us data for simulation purposes. In an actual production environment, this could be used for getting into reinforcement learning for machine learning. That's something I'll show kind of in the next example or talk about in the next example. So as this continues to run, we're able to see up at the top here, after 8 or so runs is where we start getting to optimized set points where we'll see this overall yield start going up and also become more consistent at higher levels. So again, started off with no data, took about 8 runs where the AI algorithm had to learn a little bit, kind of explore the search space, and then it was able to narrow in, start really optimizing, get into higher, more consistent yields. And when we've done this for the specific example and also for other customer projects kind of on a single unit, we've seen improvements in yield usually between 10 and 20% and reduced variability by 15 to 30% for the results that we've seen from this. And in terms of actual set points, there's different ways this can be implemented. Most of the time, what we've seen is people prefer like a human in the loop control where we're making recommendations down to an operator or a scientist then implement. But we have done, in a couple of cases, actually closed loop controls where we're directly sending the set points to the equipment. And we're currently going through the FDA approval process with the customer right now. So by the end of this year, it's expected that that'll be approved for a continuous small molecule reactor. So this was all for a single unit. Now what we can do is move on to a multi-unit, actually a multi-process unit process that we've developed a model for. So we have our upstream fermentation bioreactor. And in this case, in the schematic, we're only showing one, but it's actually a train going from small benchtop up to a 15,000 liter production reactor. Then the output of that feeds into the chromatography process. So we're kind of now at an order of magnitude level more complicated because there's way more variables. And now any change we make to something upstream is going to impact everything else downstream. And we chose monoclonal antibodies because it's estimated that over 50% of all biologics products are currently manufactured using a MAB process. So this is broadly applicable. And even though it's so widely used, there's no mathematical model to model this entire end-to-end process from the upstream all the way through downstream. And there's no optimal control strategy that's been identified for continuous MAB manufacturing either. So that's kind of what Cortex set out to change. So in a similar process to what we did for penicillin, we created a full end-to-end digital twin of the MAB manufacturing process using a combination of physics-based models and empirical data models. So using the two of those, we're able to simulate an entire manufacturing plant for continuous MAB. And we vetted it, validated it against actual production data coming out of a MAB process. Now, what does that get us, right? So now we have this high fidelity digital twin. What can we do with that? Well, the other piece that was missing for continuous MAB is that kind of control strategy. What can we do around advanced control to enable this and make sure that everything is running optimally? So we also took this model and combined it with machine learning, reinforcement learning specifically. So we're using offline reinforcement learning, actually. So we take this digital twin, we have a machine learning model, and we use the digital twin to reinforce the machine learning. So instead of needing to expose it to a lot of manufacturing data, and this was one of those limitations, right? Needing a large, clean data set, we can, with less data, we can create a highly accurate control strategy to control this continuous MAB process. And again, we vetted this against other existing control strategies not using a digital twin, so things like PID, model predictive control, economic model predictive control, and our approach using this digital twin and reinforcement learning and machine learning outperformed all of those. And we did this for this continuous MAB process. We did the same thing with that single unit penicillin. We've done it across beer manufacturing. We've done it for CSTRs and an end-to-end atropine process. And across the board, our approach to the advanced control using ML and reinforcement learning has outperformed all of them. Now you might say, okay, well, where are the numbers? How much better did you do? And that's one of the things here, is that we haven't actually implemented this in a production environment yet. We're still looking for someone who will try this with us. So if there's anyone in the audience interested in implementing this for a continuous MAB process, or if you just want to learn more even about what we've done for a single unit, or for some of the like automated PAT for the real-time quality data, or around the process optimization, any of that, there's time for questions now. Also feel free to reach out to us at Cortic. We have a booth here, booth 303. Feel free to stop by and talk to us, and we can show demos and talk through any of this in more detail as well. So with that, I think that's everything I had. So if there's any questions, I'm happy to answer them.